Today I am going to deal with the embryology. So what is embryology? The embryology is the study of formation and development of the embryo from the moment of its conception up to the time when it is born as an infant. So this is what is embryology. Now, embryology is of two types, that is the general embryology and the spatial embryology. The general one is known as a embryogenesis and the spatial one is known as a organogenesis. The total time period it takes 280 days, which is nearly about the 10 months. Now, the embryonic period has been divided into three parts, that is a pre-embryonic period, embryonic period and the fetal period. The pre-embryonic period is from the fertilization. Now, the fertilization is taking place where at the ampulla of the fallopian tube. So, from the fertilization to the second week of gestation is known as pre-embryonic period. The embryonic period starts from third week and continues till the end of second month. The fetal period starts from the 9th week to the birth. Here you can see in the interface there is a thin thread-like structure is there. They all are the chromatids, and there is a star-shaped structure which is a centromere. So this one is the centromere and these all are the chromatids. Now in the prophase we are saying that the orientation of the chromosome is taking place around the equatorial plane, and the proper orientation we can see in the metaphase. Now what will happen? Now the chromosomal arms, they will get separated and they will move towards the opposite poles. That means the one half is going to the similar pole only and the another half is going to the next pole. This is about the anaphase where they are going to the pole. Now in the telophase what will happen? There will be the not only the chromosome are already reached at the poles. So what will happen now? The cytoplasmic division is also taking place in this phase. So like this way we are getting the equal number of two cells. Division is the meiosis, and the meiosis is divided into two parts, that is the meiosis 1 and the meiosis 2. Now in the meiosis 1 they have the different phases, that is the interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and the meiosis 2 the similar phases will be there from the prophase 2 to the telophase 2. Now the interphase is taking place where? The interphase is taking place at the germ cells in the gonads, and the crossing over, which is very important because the crossing over takes place in the prophase 1. And similarly here the crossing over is taking place. Now then there is a orientation at the equatorial plane in the metaphase 1, and then the movement of the chromosome taking place towards the pole, and then there will be the separation, that is the formation of the two cell from the one cell is taking place. This happens in the telophase 1. So most importantly what is happening? The homologous pairs are separated, reducing chromosome number by half. So in this phase we are getting the chromosome number by half. Now in the meiosis 2 what is happening? In the meiosis 2 again the similar phases will continue, and that is named as a 2. Now from the 2 cells I am getting 4 cells. So I am getting the 4 cells. That means the 4 haploid gametes will be formed after the end of the meiosis 2. Now comes next, that is the spermatogenesis. Now what is spermatogenesis? The spermatogenesis is the formation of the spermatozoa from the spermatogonia. So the spermatozoa is a haploid number. You can see there is a haploid number is coming. The spermatozoa is a haploid number. So formation of haploid spermatozoa from diploid spermatogonia in the seminiferous tubule. So that is known as a spermatogenesis. Now spermatogenesis has 4 phases, that is multiplication phase, the growth phase, maturation phase, and spermiogenesis phase. Now what happens in a multiplication phase? The spermatogonia undergoes meiosis and it will produce the primary spermatocyte. it is a diploid number. Now from primary spermatocyte it will grow in size, and now what will happen, it will undergo meiosis process. The meiosis 1 is taking place, and from the diploid number the haploid number is coming. From primary spermatocyte the secondary spermatocyte will form, and the secondary spermatocyte again undergo into meiosis 2 in a maturation phase, and it will form a spermatids that are haploid. So from the two secondary spermatocytes we are getting 4 spermatids, and the process of conversion of spermatids to the spermatozoa, that is a sperm, is known as a spermiogenesis. Now next stage is a oogenesis stage, which is happening in female, and spermatogenesis were happening in male. Now what happens in a oogenesis? The oogenesis is process of formation of ovum. Now in oogenesis there is an oogonium. Oogonium is a diploid number. Now this oogonium will undergo mitosis, okay, and it will form a primary oocyte. Now this primary oocyte will develop, and its development will be arrested in prophase 1 only. It will continue as it is till the puberty. When puberty will hit, then what will happen? This primary oocyte, which was a diploid, it will undergo meiosis 1 division, and from the meiosis 1 this primary oocyte will yield secondary oocyte and the first polar body. This first polar body will die, but the secondary oocyte will again undergo the division, and it will undergo the meiosis 2. Now what will happen in this meiosis 2 division? This secondary oocyte, they will be arrested in the metaphase 2. Secondary oocyte division will be arrested in metaphase 2, and when the ovulation will be there, then at that stage the meiosis 2 is going to be completed. So meiosis 2 will only complete if fertilization takes place. So when fertilization takes place, then this secondary oocyte will undergo meiosis 2 division, and it will form a secondary polar body and a zygote, and the zygote is a diploid number. Now correspond it to the follicle development. Initially there is a formation of primordial follicle. Now this primordial follicle will convert it into a primary follicle only when puberty will hit. So puberty is hitting in a female nearly in the range of 10 to 14 years, on an average 11 year. Now this primary follicle will develop and grow into a growing follicle. From growing follicle it will convert into mature follicle, and now this follicle will rupture and it will release the ovum. When the ovum will release, the fertilization will takes place, and if there is a no fertilization, so after release of the ovum from the follicle, the fertilization takes place, and after the release of the ovum, the follicle gets sealed off and they are converted into a corpus luteum. When fertilization won't take place, then corpus luteum will persist only for 10 to 14 days, because the corpus luteum is an important source of the hormones such as the estrogen and the progesterone. As fertilization doesn't take place, so there is a no need of all these hormones, so there is a degeneration of the ovum and the corpus luteums takes place.